Okay. So, good morning all. Welcome to IWS 2020 webinar series on decovidizing biotechnology. We are starting our first session with Dr. Kshitish Acharya. Dr. Kshitish Acharya is one of the person whom I admire since the inception of my career. Dr. Kshitish Acharya did his MSc in zoology from Gulbarga University. In 1989, and he completed his PhD from Banaras Hindu University. And then he has worked under various capacities in different institutions. So he worked as a postdoctoral fellow at MRDG at Indian Institute of Science, Bengaluru, from 1996 to 1998. Then he worked as postdoctoral fellow in cell biology department, University of Virginia, USA. He also worked as head of research and production units, Yashraj Biotechnology Limited in 2002. He also founded Shodhaka Life Sciences Private Limited in 2008. And since then, he has been working as director of the Shodhaka Life Sciences Private Limited. So he is also founder of Biological Data Analyzers Association, that's BData, in 2015, which started in 2015. So he has 47 publications in peer-reviewed journal to his credit. And his major contribution is his team has identified 19 candidate marker molecules for differentiating two types of male infertility. He also has identified a specific regulatory regions that play a key role in spermatid specific transcription of ACRV1 gene. And also he has very painstakingly compiled tissue specific, database containing tissue specific expression in testes as well as uterus. So with this introductory remark, I welcome Dr. Kshitish Acharya to this session. Sir, kindly take over the session. And
okay.
It's been very nice to see you after so much long gap. It's been very nice to see you, sir. I request you to kindly deliver your session.
Thank you. Namaskar, Elrigo. So I would like to spend the next about 1 hour's time with you discussing about one of my research work. But before I begin, I would like to first of all thank the organizers, particularly, of course, Dr. Santosh, who gives me an opportunity today to share some of the research work I have done. But all the effort taken by him and other colleagues there and the department, as well as by you all who are sitting there and trying to spend some time, this will be most used if we interact a little bit more with as compared to normally what we do in the classrooms. Okay, so I request you to not hesitate when you have questions. You can ask me anytime. I'm not so much interested in just talking about the research I have, rather than interacting with you and see whatever questions you might have, if I could answer them. Okay, so it should be useful to you. Okay, so that's my intention. And I will start by sharing the slides, and you could unmute your mic and talk anytime in between. So I will share my screen now with you. I hope you will be able to see the slides.
Yes, sir.
So the topic that I'm presenting is the molecular basis of the gender disparities in smokers in lung cancer. So meaning the lung cancer patients seem to be a little bit different in terms of their physiology, men versus women. There is a difference. Is there a molecular basis for it? Is the question we asked, and I will tell you about that research. But before I begin, and as Dr. Santosh told, I'm a faculty in IBAB. If you ever come to Bengaluru, please do visit IBAB. It's a nice place to visit in terms of facilities. But of course, after the Corona break and all, don't come yet. Okay. So even our students are off the campus. We also don't go so much. But later, sometime you can come. Shodaka is a very small company, and BData is a new organization where I welcome data analysts or bioinformatics enthusiasts to come and join so that we can do some research whenever we get an opportunity together. Okay, about lung cancer. A lot of cancers do affect people in the world, and the lung cancer is one of the leading causes of the deaths because of the cancer. Okay, so this is one data that shows relatively how many deaths have been there across the globe. Things change from country to country and all, but lung cancer is a deadly disorder and we still don't have a complete cure for them. It is the leading cause of cancer death among particularly females, more in more developed countries, but it is of course affecting relatively compared to males, females seem to be affected more. And what is worrying is most of the cases are diagnosed at an advanced stage. That means when metastasis has stepped in, that is when this disease is detected. That means it is very difficult to cure at that time. That's why you have hardly 5% survivors when this disease is detected after that point. So in cancer, early detection is very important. Okay? So for early detection, what molecules we should use, etc., there is more research required. As you will see in this talk, I will present the research which actually tells maybe the diagnosis has to be a little bit different from men versus women, maybe treatment also. Okay, that's the point. That's the driving force for our research. So lung cancer, I hope you know already, it is uncontrolled division of cells. When it happens inside the lung tissue, we call it lung cancer. There are different types of lung cancer again. We'll not get into those details, but just generally treat it as lung cancer right now. And of course, more research has to be done to identify markers, as we say it, so that you can find a better way of detecting the disease early or treating better. Okay. So while there are a lot of people doing research in this area. We are also doing some research. We are using the gene expression profiles for this. And we use the technologies like microarray and RNA sequencing. So let me turn off my slideshow and talk to you a little bit about that. Okay. So I hope you can see my video, and I'm going to show you a gene. Okay. So what you see, this wire here, this is a gene, presume. Okay. And from this gene comes an mRNA. Okay. Let me put now this another wire. This is the mRNA. So from a long gene of a different length, which could go long distance with the many loops in the chromosome, a small region is used to produce mRNA. Okay, so suppose this is the small mRNA produced from one gene in the chromosome. What happens when a gene is expressed as RNA? This is finally used to produce a protein. Okay, and that protein does different functions, like hemoglobin binds to oxygen and does its job. But for hemoglobin to be expressed from its gene, there are some other regulator molecule which happen to be proteins again. So a lot of proteins do different functions inside our cells, and that is the basis of life in our cells. You know that. What genes are expressed, how much, in what cell type, and in what condition. This is critical. That's the base. That's very important to realize and understand how life happens at the molecular level and cellular level. You all agree with me. What genes are expressed and how much they are expressed. Okay. So given this situation, if you want to know any disease more at the molecular level, it is important to know what genes are expressed. Now, for example, you want to know there is a field some people are playing, First of all, you want to know how many people are playing, and among them, how many are key players. There may be some people who are cleaning the ground also. So you have to know who are the players. There may be somebody who's supplying water to the player, somebody is a physician who has come to help player, etc. But if you want to understand the game, you have to know who are the players, what are they doing, and who are the key players. There will be a lot of fielders, for example, in a cricket match, and there will be one person batting, one person bowling, one person keeping the wicket, etc. So who is the key player for that moment is very important also. It's the same way if you want to know the molecular basis of any condition, you have to know which are the genes that are expressed in that tissue in that condition. That's how it begins in terms of understanding the game of life that happens inside the cells, isn't it? So you have to, it will be nice to know which are all the genes that are expressed and how much each one is expressed. Now, what may be important for the disease is something that is expressed more in the disease condition or expressed less in the disease condition. People have techniques today to do that. So if I take a gene, mRNA, I can measure how many copies of this mRNA is present in a cell or a tissue in a given condition. For example, normal lung I take, and I have an interest in XYZ, there are techniques to measure it. Now, there are also fortunately techniques where I don't have to address one gene at a time, but find out all the genes expression so that I get a profile of all the genes. Gene 1, how much it is expressed in the normal lung. Gene 2, how much it is expressed. Gene 3, how many copies of mRNA. Gene 4, how many copies of mRNA. Like this, I have thousands of genes and their expression level. That is called gene expression profile. So I can get this for normal lung from some samples, and I can do the same thing for the lung cancer affected lung also. Now I compare the level of expression of every gene here and here, then I find that, okay, gene 1 is expressed the same way, meaning 100 copies here, 100 copies here, same level expression. There's no difference. So this may not be so important for lung cancer, I decide. If you take the second gene, this one is 100 copies and this one in the normal condition, it is 1000 copies. That means in the lung cancer, it is downregulated. There are only 100 copies of the second gene. Here it is 1000 copies. Third gene, no difference. Fourth gene, maybe it is 10 copies in the normal condition and 2000 copies in the disease condition, upregulated in the disease condition. So what is differently expressed like this? Either upregulated in the disease condition, more number of copies of the mRNA for that gene in the disease condition, or what is downregulated, less number of copies of mRNA in the disease condition. They will be important. So by comparing the profile of gene expression at the mRNA level, I can find out what are the key players. They may be very important for the disease. That will make sense in two different ways. One is to find out the molecular basis of the disease. If I know the functions already, I can understand what functions are promoted or suppressed in the disease condition. That is one. The second is I can find out potential marker list, which is a huge list by the end of such studies, but somewhere I start. So there are two benefits to compare gene expression profile in the cancer condition or any condition for that matter. I hope you understood that part. I hope you understood the significance of studying gene expression profiles. Now I told two techniques to do that. One is the microarray technology, second is the RNA sequencing technology. I will briefly tell the principle of them. What we do in microarrays, it's like we go for fishing, right? So you have a bait, you and whatever fish comes and tries to bite that, you get that, you catch the fish. But only difference is, suppose in the lake there are 5 different varieties of the fish. You are interested in knowing which varieties in how many copies. You do 10 times with one bait, which is very specific to fish species 1. How many times I get that species? I have another bait for the second species, the third one, another bait. So I can find out how many, relatively, how quickly I find different fishes. I can then tell, oh, fish variety 1, there are more of them. Fish variety 2, less of them, etc. The same way in microarray technology, we use a probe molecule that is complementary to the mRNA. If this is the mRNA, and now I take a sequence which is complementary to one small region of it, so maybe something like this. So I would have a region which is complementary. So this thread ribbon I'm trying to show is a region which is complementary to this. So wherever this goes and binds, I will actually use this as a bait to fish out this entire RNA molecule. Okay, that is the approach we use in microarray. By using this approach, by designing probes specific along the mRNA, I can fish out how these mRNA, and I can then count how many copies of each gene is there. That is microarray. I have a microscopic array of all the probes which I use to find out what genes are expressed. That means I take all the RNA, put them on this array, and whichever has more number, I will find that in more copies. That's all. That is microarray. Whereas in RNA sequencing, I take the entire RNA, all of them, simply sequence each one of them, then I actually have directly found how many copies of RNA of each type is present in the solution, which is the solution is obtained from tissues. I have to collect the tissues, extracted the RNA, and that's the solution I use. So I will, in the RNA sequencing, I directly sequence the RNA molecule. And in microarray technology, I actually make them bind to the probe of my interest. Microarray is a little older technology now. RNA sequencing is more advanced and more reliable in many different ways. So people tend to use RNA sequencing currently. End result of both these technologies is to make an expression profile and then finally find out what genes are expressed differently. So I hope you understood that. Now, let me go back to my slides. So this is time for you to ask questions. So I have told you why lung cancer is important to be studied. There are a lot of people are affected by it. There is a good amount of people affected at late stages, and many die. Very few people survive. Then gene expression profiles are critical to study any molecular basis or finding markers for any condition. So for lung cancer also. Then I told briefly about two techniques. So now about the basics, do you have any questions? This is very important for me. You understood the basics, then we can talk more about advanced topics. Otherwise, I will lose you in between. So I'll wait for questions from you. Sir, otherwise, I will do meditation for some time.
Sir, one of the questions, sir.
Yes.
Sir, somebody is asking, so what is the difference between the housekeeping gene and induced gene?
Okay, very good. So I am glad that I have a question already. May I know who's asking the question?
Sir,
was it in the chat box?
User has a number, sir.
Oh, okay. Okay. You can feel free to tell your name also, and you can talk also. Yeah. What are the difference between housekeeping gene and what was the other aspect?
Induced genes.
Induced gene. See, housekeeping gene is something that is sort of present in all the times, in all the tissues, all the cells. That is expected to be present at least. Okay, it is like, suppose you go to different buildings and find out who is there, what type of people are there. You go to a government office, you go to a factory, you go to a hotel, you go to a home. In all these buildings, there will be in police station, you go to post office. Everywhere there are people, but with different capacities, right? There is an inspector and police constable, etc. in the police station. Post office has different officials, etc. But in all these offices, housekeeping must be there. Those who are supposed to clean the house, okay, maintain the things, etc. These people are required in every office, isn't it? So just like that, there are certain genes which are essential, and they should be present in every tissue and every cell type in every condition. Otherwise, the cell will be dead if the housekeeping genes are not expressed. That means those genes which are essential, you are calling them housekeeping genes. Okay. Now something very specific to a particular tissue. For example, hemoglobin is required only in the red blood cells. That is not a housekeeping gene. Keratin is produced in the skin cells, the hair cells. Hairs are produced only in the skin cells, in some of the epidermal cells. So they are not housekeeping genes. Like that, anything that is specific to a tissue in any normal condition or disease condition, that is not housekeeping gene. That is induced during differentiation or because of some infection or some other natural disease. Okay. I hope I answered the question.
Excuse me, sir. One more question from some Anbu Ravi.
Yes.
Whether what kind of genes are responsible for cancer, housekeeping or induced?
It's normally not housekeeping genes, but our research has shown that housekeeping genes might also be altered. There are genes which are having a key function in controlling cell division. Okay, cell division has to be controlled, right? The cells cannot divide forever. So they have to be controlled. In some tissues, the cell division is always on, like in skin cells, they keep dying and new cells are produced. In some, like a lot of neural cells do not divide at all. So there is a different type of control in different tissues, but the control is there, and that control happens by proteins which are coded by different genes. So most of the genes involved in cell division control are usually the ones which are affected in cancer condition. So there are special terms for them like proto-oncogenes and oncogenes, etc. But basically this is the principle.
Okay, sir. We can continue.
There was one question, is there an age limit for cancer gene expression profiling? No age limit. If you are interested in gene expression, remember, gene expression profiling we do for any condition. If you want to understand more about any condition, it may be disease condition, it may be experimental condition, it may be age difference. For example, what happens when the skin is aging? You just want to find out, you can take a younger tissue of the same individual, maybe a mouse, for example, and then take another piece from a different region. Like this region I take from myself, let's say, in early age, around 17 years. And then at 50 years age, I again take, and at 100 years age, take another piece. I can do the study of how expression profile changed with age. So it could be any condition. In this case, we're talking about lung cancer. But obviously, what is important in lung cancer, age may be important, but what is also more important is the stage of the disease. Early stage, there is benign tumor, which is not cancerous. There is tumor, which is cancerous. And there is a late stage cancer, which is metastasis. That means from lung, it has gone out. The cells that are cancerous have gone out. That is metastasis. Gone out from the tissue and start spreading to other tissues. That is a late stage. So like the stages become important, but of course it depends on the researcher's question, what type of question they would like to ask. Okay.
Okay.
Yeah. So I'm glad we had so many questions. So that was the question of Chatina onto Louis. I do see one more question here in the chat box. I'm comparing gene expression of two different organisms, then housekeeping genes should be taken. Usually we don't consider, that becomes a comparative genomics type of study. We don't compare across the species. If you want to study the molecular basis of a disease condition, it is best to take the same species. But of course, sometimes this is also done, but that has a different purpose. But looking at the biology of a condition, we only look at one tissue under different conditions. Like I told, ages, treated or not treated, disease affected, not disease affected. This disease affected, not disease affected is the most common comparison. Okay, and we are going to do something like that in lung cancer. So coming back to my presentation now.
Excuse me, sir.
Yes.
One last question. What is the operating control in identifying gammatic gene environmental acceptance and used to fuse with during the SYNGAM I don't know. Actually, I don't
Could you
have
repeat
an
that
idea.
question? You're not completely audible.
Yeah. Now, am I audible right now?
Yeah.
What is the operating control in identifying gammatic gene and what something during the syngamy?
Now, why is that coming in cancer context here?
Oh, I don't know. Somebody has asked it in YouTube chat box.
Okay, okay.
Maybe from
See,
veterinary background, it seems.
okay, okay. See, in the cancer condition, this germline mutation and somatic mutation come into picture, but that's different compared to gene expression profiling. When you want to find out what are the key players for this cancer, there are two ways of doing it. One is gene expression profiling. The other one is mutation studies. And mutations that cause the cancer usually are germ cell line mutations. Usually, people address germ cell line mutations there. Those who are having this type of mutation in this gene are more likely to have cancer. That type of an approach. Whereas somatic cell mutations are usually used to see what happens during the disease progress. That means cancer affected cells, they have certain types of mutations. That is called somatic cell mutations. And that is important there. Now, gametes and things like that, I don't know. See, in meiotic recombination, when it happens, that's a little different concept. It is not having so much significance in cancer. I don't know. Maybe there is something missing in the question. I do perform some research in meiotic hotspots and meiotic recombination. Maybe some other time we can talk about that.
Okay.
Is there somebody speaking?
I have just muted on. Sir, 1 minute. Can you please unmute yourself?
Okay.
Okay.
Yeah. So I'm really happy that there were so many questions. So please do keep it up, and in between you can ask. But I may not see the chat box often, so you may have to unmute and talk, or talk to Dr. Santosh, and he can talk.
Sir, one question. Are there any usually observed signal that are choosing the character?
The question is not clear. Observed signals that are choosing the character, meaning.
That means many mutations are recessive, but some become dominant. Why it is so?
No, obviously the phenotype and genotype relationship is established for a lot of cases, not for every case, but a lot of cases people have observed. Okay, for example, the cancer disease susceptibility itself, there are genes available. Now, for example, you might have heard the famous incident of Miss Angelina Jolie, who's a famous film star. She had her breast removed because there were mutations in the BRCA1 and BRCA2 genes that indicated that she might have cancer later. So these are indications that she might have cancer. So she's already a mother, so she doesn't need the breast anymore. So she just got it removed.
Oh.
So like this, predictions are possible based on certain mutations and susceptibility. This is an example of marker based on the mutation. How did they identify that marker? It is based on the mutation or based on the gene expression profile. Either way, they will find that this gene is important. Then they look at the mutation of that or the molecular mechanism of it. So like this, there are a lot of phenotypes identified, including cancer susceptibility itself. And it's a well-known fact. Your eyesight or blood groups, a lot of genes have been identified. So if that was the question, that's the answer. Though the question was not very clear to me.
Okay, sir. You can continue, sir.
Yeah. Good. So then I will continue with my slides because now I will not break for your questions. But if you have questions, I can, of course, I'll be glad to take. So, okay. So lung cancer, the question we are asking, one of the questions that was that popped up in the literature was, do men and women have a different physiology? They have different anatomy, you know that, and to some extent maybe physiology because they have different hormones and all these things, you know. But about pathophysiology in terms of lung cancer, are they different? There are some indications that were coming. And because if that is yes, then maybe it's better to take different diagnosis. How do you detect the disease using what marker might be taking a different approach for men and women. That might be helpful in detecting early, maybe, who knows. Because as I told, not detecting early is causing more deaths. So if we detect early, maybe we can treat better. And treatment also itself also requires identifying markers. Markers meaning those genes which are having mutations only in lung cancer patients, or those genes which are expressed more or less in lung cancer tissue, in cancer condition. Okay. So we wanted to profile gene expression profiling. We wanted to do for men versus women, but within that we wanted to look at smoking people. That means those who, for example, here I have tried to show women and men in blue and pink color. This ribbon indicates cancer. So women who have cancer, lung cancer, and who smoke, and men who have cancer and who smoke. And perhaps more than 80% of the people who have lung cancer seem to be smokers. That's why smoking is an important aspect we should consider. So we wanted to make it very specific. So those who smoke and have lung cancer, among them, men versus women, do they have different gene expression profile? That's the thing. And in that case, what functions are affected? That's the next thing we wanted to do. So what we did is we took cancer data sets which are already published, and we try to reanalyze them by looking carefully how many samples are there from men, how many samples are there from women, etc., etc. So, and among them, how many are smokers, how many are having cancer, how many are not having cancer. So this division we had to do very carefully, and that took a lot of time actually, because you have to go to each study, go to each sample, and then compile this information very carefully. We of course also we had two types of groups. One is the men and women who smoke and who have lung cancer, and those who do not smoke but have cancer, those who smoke but do not have cancer, and normal people who do not smoke also. So like this, we had different groups identified by analyzing all the data sets where gene expression profile is already published. See, any of you can do this type of research because this is public data available. So we went to the public data. Only thing is, painstakingly for several months, we actually categorized the samples like this: those who smoke, those who don't smoke, among them how many are having cancer, how many of them don't have cancer, how many are men, how many are women, etc. We categorized them. So we actually searched for literature, and we found, not literature, in Gene Expression Omnibus, there is a database for microarray studies. We identified 13 studies where you have this type of information. There are more studies, but we carefully identified 13 studies where it is pertaining to lung tissue. There is gender information available. Study was done on lung tissue, and the information whether it is the tissue from men or women, that was available. Smoking, did they smoke or not? Patients, did they smoke or not? That was available. And were they cancer patients or normal control tissue? That information is also available. Where all this 4 type of information is available, we identified 13 studies. This took several months of research actually. It was a careful compilation one had to do, and you cannot be hurrying at this stage. So we identified different platforms and different type of studies. This is a little technical, I'll skip this. But we identified several studies and we downloaded them. We analyzed them. When we found, this is what is called meta-analysis. That means you use data sets from 13 studies and you together analyze them to find out what genes are expressed differently across men who smoke and having cancer and female smoking patients. You compared them and you found certain genes that are different. But then we filtered out those other type of genes. For example, those which are differentially expressed in men versus women, but with having cancer. That means only because of smoking you will get now. Among cancer patients, men versus women, you have removed all the genes that are differently expressed here. After removing them, you have removed those which are associated with gender bias in general. That means only among them who smoke, because of smoking only whatever genes are different that we wanted to identify. So we removed this one. Similarly, we also removed gender bias which may not be associated with cancer, because we compared normal people who smoke, and then normal people who do not smoke also we took. So whatever genes we identified different in each of these comparisons, we went on removing them. After filtering a lot of genes like this from this set, we have finally identified the gender biased genes in lung cancer, but lung cancer patients who smoke. The genes expressed differently across the genders in the lung tissue under cancer conditions in smokers. This is the final set of genes. But of course, among them, we found those which are associated specifically with women and those which are associated specifically with men. So, I mean, obviously, we have to do a lot of comparisons. So these are numbers. I will not get into the details. But when we do for analysis, finally, the interesting thing, so one is to identify the genes, how many genes are there, etc. We would identify. You will see that there are certain genes which are exclusive to lung cancer non-smokers, and you also have lung cancer smokers. So for them, you have 1,159 genes which are differently expressed. So lung cancer smoker versus non-smoker, men versus women, among them, if you see, you have 596 genes which are differently expressed, and among female smokers, cancer versus normal, when you take it, is 1,005. Like this, you get number of genes. You make a list of genes. These are your potential markers. But using them, we do functional analysis, and you can see different type of functions being regulated. And I'll come back to this in a summary. You also do something similar for down-regulated genes. This was for up-regulated genes, and this is for down-regulated genes. So those genes which are down-regulated in male smokers, they had regulation of immune process. Immune system seems to be suppressed. Whereas in women, it is opposite. Okay. So all immune related functions were actually upregulated in women, whereas they are downregulated in male smokers who have cancer. And that was a very interesting finding, actually. Nobody had reported anything like this before. So among the upregulated female smokers, the innate immunity as well as adaptive immunity associated genes were present in more number, means those mRNAs were detected in higher number. Okay, whereas in males, in upregulation, once you take, the immune related functions were not so many, so much promoted. Okay, other functions were promoted. Of course, we did protein interaction studies also to find out what makes sense, etc. I will not get into those details, but this is a heat map, what we call. So here you will see there are different genes here. Okay, each row here is a gene. This is one gene, second gene is second row, etc. And these are studies. What we try to find out here is, across studies, if I try to cluster them based on the expression pattern, do they cluster differently based on the race? Because some studies may be from Europe, some may be from Africa, some may be from Japan. So do they cluster, do the studies cluster differently? Do they group differently based on the race? The answer is no. You can see all the races are having different type of distribution. Stage of the disease also is not grouping them. Smoking pack, how much they smoke, that is not grouping them. Age is not grouping them, but the gender, if you see, suddenly you see a good grouping of female in some cases and male in some other cases. So the grouping was very prominent across the genders more than any other parameter for certain genes. And similarly, this is for downregulated genes, and upregulated genes are also behaving similar way. Means gender is the one which differentiates the certain genes compared to any other condition, any other parameter. This was the finding. To get this result, we will have to do a lot of analysis, of course. So we also have to cross-check with published literature. So we took some of those genes which are differentially expressed in lung cancer patients who smoke and non-smoking lung cancer patients, and similarly normal lung and normal non-smokers. So we're talking about gender bias in molecular markers that were already reported by the studies. What is happening to certain genes? Okay, and you see none of them have these type of pattern that we are finding. Nobody had reported. Similarly, if we look at the literature also, nobody has reported that these are potential biomarkers for this case we are seeing. Okay, So this is a compilation of the literature search we have done by different type of tools. So to summarize, there are gender disparities, gender differences at the molecular level also. And particularly we identified these 4 genes, SNX20, MTMR2, LPXN, and WIPF1. See, the other genes that I was listing here, these are the genes which are differently expressed. The numbers that I showed earlier, there are a lot of genes, right? These are the genes which are differently expressed in at least 2 studies. But is there something that is differently expressed in all the studies? Yes, these are the 4 genes. In at least 5 studies, no, not all the studies, 5 of the 13 studies. That was the highest we could get. There's nothing consistent in all the 13 studies. But at least 5 of the studies, if there was something consistent, that was the highest number we could get, that are these 4 genes. And more importantly, differently expressed genes, when we look at it, indicates that immune responses are elevated in female smokers. That's the indication. Okay, so those are the highlights of the study. So this is a good example of how you can make use of the existing gene expression data, which takes a lot of money and time to collect this, identify the samples, meaning the patients and the donors, and then collect the tissue and then do the study by putting a lot of money into sequencing experiments and then analyze them. You already have data from the public repository, so we made use of that data. Of course, we did somewhat patients testing job in filtering them out separately, identifying each data set for the certain parameters, and then selected this certain studies, 13 studies, and we analyzed after downloading the data. And this is the result. When we started the study, actually there was no such report at all. But after that, studies came out, so we have a little bit of a challenge in publishing this now, because it has, though it was the first discovery, we presented in a research seminar, but after that we couldn't publish it because of certain reasons. We're a small team, so there was a delay in publication, and now some similar results came up. Actually, smoking only we took because of that reason. Otherwise, molecular basis of gender bias in general in any lung cancer condition, men versus women, when you take smoker, non-smoker, all this, we were the first people to show actually, but we did not publish it. We're first people to find, but not the first people to show it. So it is very important to be fast in publications. Anyway, that's another lesson that you should always keep in mind. Yeah, that's it. So that's the summary. I should definitely thank again the organizers, and 2 important contributors of this research are the people marked there. Akhilesh, Mr. Akhilesh and Mr. Ravanti are my PhD students, and they're fantastic actually. So they have not just done the analysis, even in designing of the experimental conceptualization, etc. They have a very important role. Okay, so they are not like very passive. I tell them to do, okay, you do this, they do it. It's not like that. They also contributed in design of the experiments, in literature search, reviewing the literature. In a lot of different ways, they took huge role. They contributed hugely to this research project. So I contribute them. Of course, my team is having different contributors at different time points, as you can see there. That's it. So now, next, more questions from your side, if you have understood the talk.
Thank you, sir, for a well-informed talk. I have one question from Dr. Anant Krishna. Hello.
Yes, please. Yes, please.
Sir, can you hear me?
Yeah, I can hear you.
Right. Does the woman exposed to firewood cooking smoke for longer time, whether they are at the risk of developing lung cancer?
That is definitely a possibility. Some research has shown that those who are prone to cooking have more lung disorders. They're more prone to lung disorders than other people. Even the women itself who are not exposed to that versus this. There is a study done, but not so many studies have been done like this. What we have done. We can't comment on lung cancer of different stages and all. But in general, yes, lung cancer disorders, the lungs are more harmed, both by people who are exposed to this cooking by the traditional method, who actually used to, particularly in older kitchens, you might have seen our older houses had very small windows. So, and women will be blowing, and then the smoke will be coming, there will be tears in their eyes, and it is manageable, so they manage. But eventually the lungs would be damaged to some extent. Of course, our lungs have a mechanism to repair itself, but the damage would be there, and there are higher chances of them getting the disease. So one interesting aspect I would like to tell here, pollution also has the same effect. Okay, pollution in Bengaluru, cities like Bengaluru and all, we have more problem actually. In fact, cardiac arrest possibilities are also higher because of nitric oxide pollution, which is odorless. You don't even see that as a dust or anything, but that is one deadly thing. Anyway, coming back to the point, um, when you have any type of this type of contaminants, etc., our body has a way of repairing and all that, and that's why you don't see every smoker getting cancer. Fortunately, 90% or more than 90% of the people who smoke do not get cancer. Those who eat tobacco, they don't get cancer. But don't use that logic to start smoking now or continue smoking, because 90% or 80% of 80% and more of people who get lung cancer are smokers. So your choice. You want to be in that 80% category or not? Do you belong to 20% of the people who smoke and don't get cancer? We do not know. So it's a large number of smokers, but those who get lung cancer, 80% of them are smokers. So like this, it might be the case for women also, but detailed studies are required.
Sir, one more question. Is the intensity and life expectancy also has a gender dependency in lung cancer?
Yeah, that was already shown. In fact, women are affected more. There are more men who are affected by lung cancer in general in the world. But among those who get the percentage, if you see, women are affected more. Their progression of the disease is faster and deadly there.
The four genes that you concluded that gender bias in lung cancer. So all the four genes are related to immunity?
Not directly. Actually, they have not so much. So I have listed the functions in the bottom. You can see one is the first one is SNX20 is a protein that binds to phosphatidylinositol lipids, and it interacts with the cytoplasmic domain of PSG1 and cycles the PSG1 into endosomes. This is what is known. So like this, there are different functions. None of them have any direct correlation with immunity. But when I take all the group of genes which are upregulated as a group, it seems that the pathways and gene ontology functions that are associated with immunity are upregulated in women, unlike men, where they're downregulated.
So one more question from Triveni.
Yes, please.
I am going to add to that question also. She is asking about chances of lung cancer in passive smokers, and also whether you can implicate the same genes in passive smokers also.
Well, the second part I cannot speculate on that, because studies have to be done, but possibilities are there. See, passive smokers or active smokers, actually there seem to be two types of people among all of us. Those who can metabolize external unwanted material that goes into our body, some of us can metabolize it faster. Some of us can't. And that's the key difference actually. Passive or active, it doesn't matter. The amount of smoke that goes in and how susceptible we are. So the lung doesn't care whether I'm actively smoking it or passively somebody is smoking and I'm getting it. It doesn't matter. So for some people, even a small amount of smoke can damage a lot. For some people, a lot of smoke is required to damage the tissue. So there are different parameters that play a role there. So we will not be able to straight away make a conclusion, but yes, it is shown that passive smoking also is equally bad. It can also be dangerous. That is shown.
Sir, whether these some Nagavan is asking, Vasista is asking, what are the chances of getting lung cancer in people related with textile industry? It is there or not? No.
Any pollution, including textile industry dust, can potentially cause the problem. But I do not know if any particular studies have been done. So somebody has to do the study. In science, it is very important, because I'm sitting here as a scientist and you are you are giving me this privilege of being listened to. I shouldn't say the wrong thing. So where the studies are not done, we should not tell anything. So I am not aware of the studies that have been done in this context. So somebody has to do the study. For example, you find out all the people in the textile industry take certain this thing. Some of you can do that survey actually. How many of them are affected by lung cancer? And compared to an equal number of general population screening. General population, anyway, it is already available because you can also screen. Suppose you take about, to begin with, 100 people in the lung cancer, those who are going to garments, etc., textile industry, working there. You take 100 people and take 100 people of equal age group from a normal population who do not work in textile industry, and you can compare. But 100 is a small number, you have to do for thousands. But I'm just saying how that would be done. So some studies are done, then only we will know exactly what is the situation. But theoretically, yes, more dust, more pollution can actually damage the lung tissue and can cause lung cancer also.
So one more question. Whether your survey included, somebody is asking, whether your survey included the specimen or the results, whatever the geo you accessed, geo profiles you accessed, whether that had a condition where lung cancer is due to the metastasis of a cancer present in other organ.
No, we did not. We took only lung tissue.
Okay. So
because then it becomes comparable. Otherwise, it's very difficult to compare. And not so many tissues were available, because when you talk about other tissues, metastasis, there will be multiple tissues in different, when different tissues may be considered in different studies.
Okay. One more question is that the four proteins that you mentioned at the end, whether they are giving the drug resistance,
Whether they are giving
chemotherapeutical resistance to chemotherapeutical.
We don't know. We don't know.
No.
They have to be studied. So that's the whole point. Our point is
they are the candidates who are predisposing to lung cancer.
No, we do not know that also. Okay. So what we have identified is 4 genes that are consistently differently expressed in 5 of the 13 studies. What does that mean? Then there is nothing like 100% in 30, all the studies in all the women it will be there. But then there may be, we have to now go back to that hundreds of genes and find out whenever gene A and B are upregulated, that is one type of group. C and D are downregulated, that is another type of group of people. So like this, we might have women who can be segregated among the patients. We can perhaps segregate them in different bins based on the type of genes which are upregulated and downregulated. That means there are not only men versus women are different, among women there are different types. So what are the genes to be studied further to actually go there? This possibility exists is what we have shown. There are 4 genes we have shown that is most common. There were 9 which were actually at least 4 of the 13 studies, like that. The number increases, and if you have at least 2 studies, then you have about close to 1,000 genes. We have to analyze them and perhaps even take up more samples and do the studies in terms of specific combinations of them coming differently. So we can group or cluster women with lung cancer among the smokers. We can group them differently based on the gene expression patterns and what genes are categorizing which type, etc. This can be studied. That possibility is shown by our study. Okay. So most studies are like this. They give a clue for the next study.
Sir, one Varsha is asking how effective it is to find out the molecular basis of lung cancer in successfully rescuing the patients from lung cancer. Whether it is going to promote the treatment regime or what?
So, when we talk about helping the patients, we talk about 2 things. One is the diagnostics. You have to detect the disease early. Second is you have to treat the patient with some drug. To detect the disease, you have to identify potential markers, which genes you would use to detect. So our study is a effort in that direction. What could be the potential markers? We have told for the first time, look, don't try to treat men and women equally in this research. Maybe you have to treat them separately, number one. Number two, even among them, there may be subgroups of men and women who actually have different type of gene markers. So you have to use a panel of markers, either this type of genes or this type of genes or that type of gene for diagnosing. So if you use, eventually, if you develop a better diagnostic kit like this, you may be able to detect the disease earlier and thereby saving the patient's life. Your early detection itself can be saving the patient's life by several folds. So for detection, that's the first place where our research is relevant. The second is when you want to treat, how is the treatment work? I think most of you perhaps know this. There are small molecules, what we call drugs, which go and bind to certain proteins. And when they bind to the small, bigger proteins, their activity changes or their activity stops. And that helps in curing the cancer. But what is that protein you would like to choose? Some of these genes, what we are listing, not the 4 only, there are thousands of them. Among them, you have to do more research and try to find out, they become the targets for treatment later. Targets for research for treatment.
Okay.
So we would have shortlisted like that. So eventually we will perhaps get there. This is how research happens.
One Swati is asking, how do you think this analysis would be helpful for early diagnosis of lung cancer in different gender? Is it based on only those genes or, or that are different in male and female?
Yeah, I think I answered that question. So the fact that there are about close to 1,000 genes which are expressed differently in men versus women, we have now segregated men associated genes and women associated genes for lung cancer, as far as lung cancer is concerned. Among them, we have segregated those who smoke and don't smoke. So a doctor could ask, for example, imagine a situation if this research continues in this line. A doctor could be asking, okay, first see, okay, whether it is man or woman. Okay, if you are a woman, then do you smoke? Then you have a sub list of genes which are identified. Maybe there are 4 or 5 groups of genes. You have to screen for expression of those genes in the blood eventually. Right now, this is in the lung tissue. You have to now do a search them for the blood, or if there is already a lung thing, they actually take a small biopsy and test the lung itself. Either way. So that's how research has to be done. But to do that next step in the research, we have given results. But it won't be only these 4 or 5.
There is a question which is slightly bit deviating from your topic, but since you are making YouTube videos on awareness of COVID, I felt this to be asked to you. Okay. For your information, Dr. Kshitish Acharya is actively involved in preparing the video for COVID awareness in native Kannada language. So I request you to go to his channel and view all those videos, which will give you, which is very, very informative. You won't regret going there, I'll tell you. Sir, one question is, if smoking person comes in contact with COVID patient.
What person?
If a smoking person, if a person who is smoker.
Smoker, okay.
Okay, yeah. If he comes in if he is a primary contact of a COVID person, right? Does he stands a chance to survive?
The theoretical prediction would be those who smoke are more susceptible to any lung damage.
Okay.
And COVID-19 affects the lungs, so smokers should be more careful. So that's definitely there, but nobody has done a study to establish a correlation like that. Okay, so we can't say for sure that this is how it will happen. So again, thanks Dr. Santosh to bring up that point. So I have written at the bottom, simple straight science in Kannada. So there is an email based on this, S3 in Kannada, s3kannada@gmail.com. So you can connect me on Facebook. I will keep publishing some of my these videos. And one of the points I made in the first video I made is, in science, we should not, sometimes when students ask question or laymen ask question, you might be facing somebody, one of your relatives asking question, you are studying a science, what is your opinion? When somebody asks like that, a lot of times we become immature. We just answer something as if that is the standard rule. So you have to be a little careful. For example, this question, theoretically what is possible is very different from what is proven. And we have to state the fact. So there's no evidence that this happens. Nobody has studied, I think. So it's a good thing to study, but in theoretical basis is what I told, that it is possible that those who smoke are more likely to be having higher danger in COVID-19. However, please do remember again, of about 100 people tested, 15 test positive. Among 100 people who are tested positive, 97 people, nothing happens to them. Okay. So far the focus is more on some other complications where 3% of the people have severe problem and they might even die. So they're smoking and non-smoking, perhaps, I don't know, among those only, it's a very small sample. People have not studied it yet.
Sir, one question from Kiran Nagwan. I am going to reframe it for you for better understanding. The candidate gene that you have identified, which are the risk factor for developing lung cancer. So can they be controlled by? I mean, can the what do you call the gene therapy can be an answer for rescuing them from the lung cancer?
So whenever we identify certain things, see, those 4 are more interesting because they are present in 5 of the 13 studies, but they are not present in 13 of the 13 studies. So you already know that they are not the final answer, number 1. Even if suppose I have found 2 genes or even 10 genes or only 1 gene that is downregulated in all the 13 studies, does that mean your research has ended? No. Our research only has told that there is a correlation between the downregulation of this gene and the cancer or the smoking, men and women would say, whatever it is. That correlation, is it causing the disease? Can I use this to detect by testing the blood? Can I use this as a target to treat the disease? All these questions have to be addressed now by doing further research. Okay. So what we have done is, by making use of the existing gene expression data, we have shortlisted potential markers which are more promising, which have to be studied further in various contexts. That's how research happens. Correlation is not causation also.
Okay. Sir, a person, I mean, actually his ID is coming like AJ. Sir, my question is, the people from desert are more exposed to the dust. And whether the exposure to dust, whether they get immunity to fight against lung cancer, or it will make them more prone towards the lung cancer.
Anything, the dust-like material that damages the lung is not good for lungs. And smoking, of course, is even worse because there are chemicals, not just regular dust. So they are not good for lung. And whenever lung is having some problem, there is a higher chance, slightly higher chance of them affected by lung cancer. But that alone, none of these alone cause cancer. Smoking alone doesn't cause cancer. Dust alone doesn't cause cancer. Pollution alone doesn't cause cancer. So same thing about the deserts also. In desert, the dust is not always there. It is sometimes there, not always there. But generally speaking, dust per se, that alone doesn't cause anything. It damages the tissue. So if the person has a higher susceptibility because of the other genetic predisposition, there are certain families who have cancer more. So if there is something like that, if there are certain mutations in some genes or their lifestyles is not good. So in shodhaka.com, if you go, I have tried to make a small panel, how to prevent cancer or any disorder for that matter. There are some simple things about how your lifestyle is. There are certain populations who never have cancer, certain tribes and things like that, for example. It is not only because of their genetics, but it is also because of their lifestyle. If you are working most of the times, if you are acting and you are not eating too much, you're not a person who eats for the sake of eating. So probability of you getting a cancer in early ages is very less, unless you are exposed to some chemicals and things like that. So my point is, while any lung damage has a higher chance of you getting a lung cancer, that doesn't mean just because I have some damage to the tissue means I will get lung cancer. The possibility becomes higher. The other outlying factor is other undermining factor is very important, which is your lifestyle and your genes. If you're already susceptible for because of these things, now the chances of an average person and you who is exposed to certain parameters which damage the lung in general, smoke or dust, your chance becomes a little higher among them. That's all.
Sir, whether asthma predisposes to lung cancer?
No, there is no evidence for that.
No. Looking at the genes involved in lung cancer, does it suggest that it has a genetic predisposition? One more question.
Yes, there is evidence that that indicates that there is genetic predisposition for many cancers.
Sir, in general, whether it is always necessary for the cell cycle controlling genes to go rogue for cancer, or are there any other proteins also which can influence on development of cancer?
Cell cycle controlling genes are very important. Now the network keeps spreading, right? So when you have cell controlling gene, when I say, so protein interactions, that's why they are important. Suppose this is a directly known cell controlling genes, but its phosphorylation is this may not be known yet. And there may be some other transcription factor which is regulating that cofactor. So like this, the cascading goes on. So finally, cells controlling proteins that matter and those which influence that protein. So you can again call them also self-controlling proteins. However, but please remember, when we say cell division control, there are some which have to be high to control it. There are some which have to be low to control it. So up or down regulation, both might be deadly, depending on what gene we are talking about.
Sir, in your experience, whether the nicotine present in the smoking, whatever the smoke material, whether that has any direct effect on pancreas also other than lungs?
I have not read the literature in that direction.
Okay. So this study was more focused on lungs only, right?
Yeah. This study was more focused on lung cancer and not even lung cancer pathophysiology or the chemicals associated with smoking. It's just pure study of lung cancer pathophysiology in men versus women at the molecular level, based on gene expression profiles.
There is a question connected to your previous answer, where you said anything that goes into the lungs causing the problem has a potential risk of developing cancer or something like you told.
No, it enhances the risk to some extent. By some fold, that possibility is there.
Now, taking that thread, Malik is asking, sir, nowadays due to COVID, everybody is asking, everybody is wearing mask. Whether this kind of wearing the mask and avoiding, whether it is going to affect the onset of the lung cancer condition or what?
The masks that have plastics particularly might raise this question, but very unlikely. But we never know for sure. One has to do a study on that. But as I told earlier, any chemical that comes in our body, whether it is through the respiratory tract or by food, whatever, or by skin absorption, our body has some capacity to deal with them. So don't worry if small amounts come in. Okay. As I told in the beginning, passive smokers, when I was answering that question, I was addressing that there is always a scope that something comes in. To some extent, our body can tolerate. That toleration ability changes from person to person. Now we do not know that. So it is better to minimize anything that is foreign into our body, including food. Food also, just because you can take it, if you take too much, there are problems. Any food. Nowadays they are talking about vitamins and things like that, this kashaya and all that. Don't do anything. Visha, Amrutanu Vishati Adra Vishanta Heltare. So be cautious. Is my suggestion. And mask also, you should wear it when required. You see, what is the benefit of mask? I have put that at the bottom. If you search in Facebook Kshitish Acharya and you confirm that I am from IBAB, you will get my note. And there I will update my videos. In one of the videos that are coming up, I'm going to tell you, most people miss the benefits of mask. Mask is having two effects. One is it actually prevents, if I'm infected, from me it doesn't go out, and from outside it doesn't come to me easily. It can still go out, it can still come in, but it doesn't happen easily, number one. But the second important benefit, whenever I'm wearing mask, I'm not touching my nose and mouth with fingers because there is a block. So that is important too, because you have, before you wash the hand, you have some time, otherwise you will be unconsciously touching it. That's the second benefit. Now, because I'm wearing the mask too much, if there can be a problem, I don't know. Somebody has to do a study on that. Probably no.
Sir, through your analysis of gene expression, did you came across any genes which confers the resistance to the cancer? I mean, what he is trying to answer is whether you have come across the genes which can confer cancer against the treatment regimes, whatever they use,
Well,
making the
some
cell
of
to
the
go
genes
rogue.
which are well studied are already known to have role in fighting the cancer and all that. So there are many, obviously oncogenes, when they are expressed differently, they are known to have some such effects already. So there are many like that. But what is interesting to us is that there are, for example, there is this gene, these two genes. Nobody has studied and reported in any context. That completely new genes. There is one here also. Okay? So when you look in the literature, there is absolutely no evidence for some of the genes. This is some example only after thousands of genes we have studied. So maybe those are the interesting cases to explore further. Otherwise, there will be quite a few genes which are known to be associated with lung cancer and resistance to lung cancer and all that.
So one general question is being asked to you, sir. Whether the biomarker and the drug target, they can be same or they are different altogether?
Biomarker is what? Biomarker is a gene or any molecule that is expressed differently or present in a different shape, etc., in the condition 1 compared to normal.
Okay.
So that becomes a marker. If that gene being expressed more is a marker for that disease, maybe. So we call it a biomarker. That gene being expressed less could be a biomarker. That mutation could be a biomarker. So marker is simply association in a different way with the condition. That is, of course, that we now say potential marker. When we prove with more samples, we call it a real marker. From the real marker, now how do I make use of it? Can I use that to diagnose the disease or to treat the disease? Both possibilities exist. There's a third possibility. You understand more about the molecular basis simply. For all the three cases, we can focus more on biomarkers, and that's why biomarkers are important. What is the real application? Only more research will tell. Biomarkers are help. Discovery of biomarkers will help scientists to focus better on certain molecules. That's all. In all the three directions I told: diagnostics, therapeutics, treatment, and basic research.
Okay. From Neha Tabasum, people with pneumonia have a chance of developing cancer, lung cancer. Can they develop lung cancer?
I don't know. Maybe not. It may not be affecting so much. But pneumonia itself is a deadly disease, so you don't have to perhaps wait for cancer. You have to deal with pneumonia separately.
Okay, so that's all the questions, sir. And I think we have come to the flag end of the session.
Yeah.
Because I'm
But
not
I should
getting.
tell Dr. Santosh, I should congratulate you also, as well as your students, because I have not seen this much frequency of questions normally. So we have to prompt them a lot. So I think you have prepared them well. So thank you very much. And thanks to all of you who have
No,
asked the
one thing I have told, I didn't told them anything. Number one.
Okay.
Number two is when you asked, then only I put it in a chat box that you please ask a question, if any.
No, that was wonderful because those interactions are important, and I'm glad so many of you asked the questions. So those who asked the question and those who otherwise listened also, thank you all of you. And if you're interested in my videos that I talk about science factors, actual facts in science, I would like to try to tell them in Kannada in simple terms. Please do connect me on social network, and I'll stop here.
Sir, at the outset, I'd like to thank you and I express my deep sense of gratitude for spending your time with us and enlightening our students with various aspects. It will be more helpful for me and it will be benefiting the students if you could talk only 2 minutes about the things that are going at IBAB and how one can enter into IBAB for pursuing their career.
Okay, thank you. I think it is very important aspect that I wanted to talk also. IBAB is a very different type of institute for India. A lot of new things are happening, and I joining IBAB. One of my first PhD student, actually not one of my first PhD student, happens is from Coimbatore University, Mr. Darshan. He was a student of Santosh sir also, and a very good student. He's now in University of Alabama. For PhD or for MSc, we have very good facilities, and if any of you are enterprising, you should definitely try. The competition is tough. About 500 to 1,000 people apply, and 30, 40 get selected. But if you are interested in science, why not, right? So you should try definitely joining IBB. Even otherwise, if you visit Bangalore, please do come to IBB. I'll try to show you around later. I also give career planning talks and things like that, and I'm interested in, if some other time permits, I would like to talk to you separately, both about IBAB or in general, how to plan career, how to plan for higher studies, whatever I know a little bit, I would like to share if that happens. But you please visit IBAB website at least, whereby you will see how different faculty members are doing different type of research, and you will get a glimpse of it. There are some videos also available online, but it's a good place to be. I think all over India, if you look at the placement record in life sciences, it's around 25% in life sciences. But IBAB has a record of close to 100% from the first batch so far. In 2001, we started, and from then to now, all batches we have, all different courses we had, of course, everywhere we had achieved that. And that's because of the quality of the students in the first place, and also the type of network we have, the facilities we have, all of that matters. And so it's a special place that way. Not many places in India have achieved that. So, and we used to put, now I don't think we are putting, because now it's sort of a small thing for us. Where earlier we used to put every batch which student is placed where. Okay. Now we're not highlighting that so much, but it is the record is still like that now also.
Okay.
But I'll not tell more because I don't want to take too much time from you.
Okay, sir.
Some other time
Sir,
I can tell more about the career planning also if there is an interest.
we are planning also to make a special session like a talk with an expert, an half an hour session followed by interactions by the students. So
Sure.
that is in the pipeline actually. And the moment we get a nod from the university, we will be doing that every day. I mean, every week one session will be there. So we are already mapping some important resource persons whom we can approach, and we can oblige our approach also.
So again, when it is organized, please participants, please make use of it, because I'm sure there will be others also who will be speaking. If how much you are prepared will help to actually make use of that session. For example, in the videos that I'm preparing to make after the COVID-19 series, one of the first thing I want to take is basics of career planning. And maybe with that preparedness, it will help to interact with others also.
Okay, sir. Thank you for being with us for somewhere around 1.5 hour and enlightening our students with your knowledge and also the career aspects in our field. And hope I will get your support in future also, sir.
Oh, anytime, please. It has been always a pleasure interacting with you, Dr. Santosh, and of course your students and your team members. So thank you very much.
Thank you, sir. Thank you very much. So that's the wrap up of our first session of IWS series. So please stay tuned to the YouTube channel. We'll be back by tomorrow 10 o'clock, where Dr. Bharat is going to talk about perspectives and prospects of computer-aided drug designing in the COVID era. So he is a research head in Atrimed Pharmaceuticals, who are currently involved in Ayurvedic and herbal-based medicine. Also, he is the head of computational chemoinformatics division also. So recently they have come up with some 3 molecules which have shown very promising results against combating COVID. So stay tuned, and we'll bring him for more information about his research also. So I thank you for being with us. Have a nice day.